Hello everyone, hope you're all doing fine. Today's topic is complement activation pathway, and I'm going to take it with the help of PPT of my dear colleague Farzana K. Mohammed. So complement activation pathway, you want to know about this because it is really important topic in case you're studying immunology. So complement refers to a system of factors which occur in normal serum and are activated characteristically by antigen-antibody interaction and subsequently mediate a number of biologically significant consequences. So complement have the components or factors. Complement factors is really glycoproteins, okay, and that is really seen in normally in serum, and it is seen really inactivated. So it should be activated characteristically by antigen and antibody interaction and subsequently mediate a number of biologically significant consequences. So its function, when we say about its function, lysis of cells, bacteria and viruses, opsonization which promotes phagocytosis of particular antigen, and immune clearance which removes immune complexes from the circulation and deposit them in the spleen and liver. So this is an important part of immunology or immunity in our body, that it will clear the immune complexes. It will lyse the cells and bacteria, it will do the opsonization, and it will promote the phagocytosis. We know how the phagocytosis happens. Normally it is done by macrophages. The white blood cells, monocyte, will differentiate into macrophages and it will undergo phagocytosis. Phagocytosis, we know that phagocytosis is an important part of innate immunity, which is less specific. Innate immunity is working with 4 kinds of barriers, which is anatomic, phagocytotic, and physiological and inflammatory barrier. So one of the barrier is phagocytosis barrier, that it will clear the invaders by engulfing that. So lysis of cells, bacteria, viruses, and opsonization, phagocytosis, everything came into the function of this complement system. And the general properties of this, C means complement, is present in sera of all mammals. The proteins and glycoproteins that compose the complement system are synthesized mainly by liver hepatocytes, but significant amounts are also produced by blood monocyte, tissue macrophages, and epithelial cells of the gastrointestinal and genitourinary tract. So the complement system have some factors, that is like proteins and glycoproteins, and that is synthesized from the liver, mainly by liver hepatocytes, and some amounts is also done by blood monocytes and macrophages and epithelial cells of gastrointestinal and genitourinary tracts. So how much it constitute in normal serum proteins? It constitutes 5%, and it is not increased as a result of immunization. Most circulate in the serum in functionally inactive forms are proenzymes or cymogens, which are inactive until proteolytic cleavage. Not inactive, it will be active after a proteolytic cleavage. Until it will inactive, as we mentioned here in this definition. So it will be inactive and it will be active only by proteolytic cleavage. And it is heat labile. It is denatured in 30 minutes at 56 degrees Celsius. And the another properties of this complement, it does not bind to free antigen or free antibody, but it only bind to this antigen antibody complex. And C with bound Ig activate classical C pathways, that we are going to say it upcoming section, because this complement pathway can be happen in 3 ways. Classical alternative lectin pathway. So when the C is bound with Ig is an immunoglobulin with a gamma heavy chain class. So when it is bound with Ig, it will activate classical C pathway. All classes of Ig do not fix the complement. Only IgM, IgG3, 1 and 2 fix complement. And the site of C binding is located on the Fc piece of Ig molecule and is expressed only when Ig is combined with its antigen. The fixation of C is not influenced by the nature of antigen, but only by the class of Ig. So next we are going to say about the complement components. So we already say that complement components are like glycoproteins and so and so. So complement system consists of at least 20 chemically and immunologically distinct serum proteins. Complement is of complex of 9 different fractions such as called C1 up to C1 to C9. Peptide fragments formed by activation of a compound or component are denoted by small letters. The larger fragments bind to the target near the site of activation, and the smaller fragments diffuse from the site and can initiate localized inflammatory responses by binding to specific receptors. The complement fragments interact with one another to form functional complexes. So this is all about the complement components. And complement components can be classified into 7 functional categories. Before it, a scientist from Berlin, Paul Ehrlich, defined this complement system as the activity of blood serum that complies the action of antibody. So now we are going to say about the complement components, which is classified into 7 different functional categories. So it is mainly proteins and glycoproteins, which is produced mainly from liver hepatocytes. So first category is initiator complement components. So as the name indicates, it will initiate the complement pathway. So initiate their respective complement cascade by binding to the particular soluble or membrane bound molecules. Once bound, they undergo conformational changes, resulting in the changes in their biological activity. So example is C1q complex, mannose binding lectin, and ficolin, etc. I don't think there is a need of further explanation of it. So next category is enzymatic mediators. Several complement components are proteolytic enzymes that cleave and activate other membrane members of complement cascades. Some of them are activated by binding to other molecule and undergoing a conformational change. Others are inactive until cleaved by another protease enzyme and thus termed zymogenes. Example C3 convertase, C5 convertase, C1R, C1S, MASP2, factor B. And the third one is opsonin. Upon activation of complement cascade, several proteins are cleaved into 2 fragments. Each of them takes on a particular role. For C3 and C4, the larger fragments, and C3b and C4b serves as opsonin. They enhance phagocytosis by binding to microbial cells, serves as binding tags for phagocytic cells bearing receptors for C3b and C4b. Before I say about the opsonin, we want to know about the opsonization, which is playing an important role in immunology or immunological response. Opsonization is called opsonization is the promotion of phagocytosis of antigen by macrophages and neutrophils. Antigen and antibody will form a complex, and it will have a Fc fusion receptors antibody, and that is called opsoning. So that is due the phagocytosis of antigen. It's an important thing in immunological response. So next is inflammatory mediators. Inflammatory mediators that enhance blood supply to the area they are released by binding to the receptors on endothelial cells lining the blood vessels and inducing an increase in capillary diameter, attract other cells to the site of tissue damage, called anaphylatoxins, causes anaphylaxis. Example for this is C3A, C5A, and C4A. As I said before, the immunity have 2 branches, that innate immunity and adaptive immunity. The innate immunity works with the 4 barriers, and one of the 4 barriers is inflammatory barrier. That when a wound or an infection happens in our body, a series of programs will happen, and that is called inflammation. And the 4 cardinal signs of this inflammation is explained by the scientist Celsus: redness, pain, temperature, swelling. And the scientist Galen have added one more point: it is loss of function. So inflammation should clear the invaders from our body, either by innate or adaptive system. So for it, this inflammation, when inflammation happen, 3 events will happen. One is vasodilation. That is, in damaged region, blood capillaries will enhance in diameter, more blood will flow, more red, and more temperature will occur. And the second event is capillary permeability. That is, capillary wall becomes more permeable, so more fluids will come to the damaged sites. Okay, and then third event is influx of phagocytes from blood to tissues to do the phagocytosis of the invaders. So this is about the inflammatory barrier which is in the innate immunity. So I think there was a need to say about this inflammatory barrier before I saying this inflammatory mediators. Okay, and then the next, membrane attack proteins. They insert into the cell membranes of invading microorganisms and punch holes that result in the lysis of pathogen. The example of it is C5b, C6, C7, C8, and multiple copies of C9. And the sixth one is complement receptor protein, which is present on cell surface, bind complement proteins and signal specific cell functions. Its example is given below. And the seventh one is regulatory complementary components. Example is factor 1, degrades C3b and protein, inhibits the formation of MAC on host cell. MAC is membrane attack complex. So next we are going to start about this complement activation and the complement activation pathway. There is 3 pathways. So it can be divided into 3 pathways: classical, lectin, and alternative. All of the 3 result in the activation of C5 and lead to the formation of the membrane attack complex. The 3 pathways differ in the way C5, complement 5, is broken down, but after the formation of MAC, membrane attack complex, is essentially the same. Initiating event of 3 pathways are different, but converge in the generation of enzyme complex capable of cleaving C3 molecules to 2C3A, C3B by C3 convertase, and second convertase get C5 convertase cleaves C5 into C5A, which is the inflammatory mediator, and C5b, which is the initiating factor of MAC. So first we are going to say about the first pathway, classical pathway. The classical pathway is triggered primarily by immune complexes in the presence of complement components 1, 4, 2, 3 and calcium and magnesium cations. C1 is the first component to participate in the classical pathway. It is composed of C1q, C1r and C1s. I think now you get an idea about the things we have mentioned before. We have mentioned complement components. So I think C1q, C1r, C1s was there. So now it makes sense, I think. So C1 is the first one to participate the classical pathway, and it is composed of this C1q, C1r, C1s. The C1q reacts with the Fc piece of bound IgM or IgG. C1q binding in the presence of calcium ions lead to the sequential activation of C1r and C1s. Activated C1s enzymatically cleaves C14 into C14A and C14B. Not C14A, C14A, C14B. So the activated C1s enzymatically cleaves C4 into C4A and C4B. C4b binds to the antigen-bearing particle or cell membrane, while C4a remains a biologically active peptide at the reaction site. C4b binds C2, which becomes susceptible to C1s and is cleaved into C2a and C2b. C2a remained complexed with C4b, whereas C2b is released. C4b to A complex is known as C3 convertase. C3 convertase in the presence of magnesium cleaves C3 into C3A and C3B. C3b binds to the membrane to form C4b2A 3b complex, whereas C3A remains in the microenvironment. C4b2A3b complex functions as C5 convertase, which cleaves C5 into C5A and C5B. Generation of C5 convertase marks the end of classical pathway. C5b initiate the formation of membrane attack complex, MAC. The formation of MAC, C5 convertase cleaves C5 into C5a and C5b. C5b instantaneously binds C6 and subsequently C7 to yield a hydrophobic C5b67 complex which attaches quickly to the plasma membrane. Subsequently, C8 binds to this complex and causes the insertion of several C9 molecules. The insertion of membrane attack complex causes formation of a hole approximately 100 angstrom in the membrane, thus lysing the cell. So the next is alternative pathway. Alternative pathway is so called because it bypasses the requirement of antigen antibody complex C1, C2, C4 complex. So, because in classical pathway we have used complement components 1, 4, 2, 3 and calcium and magnesium ions, where this alternative pathway only or doesn't need antigen antibody complex C1, C2, C4 components. It starts with the spontaneous activation of C3 in serum and requires factors B and D, magnesium, all present in the normal serum. So it starts by the activation of C3, which is already present in the serum, and also requires the factors B and D and magnesium, which is already found in the normal serum. So it doesn't need any antigen antibody complexes or C1, C2, C4 to start it, like the classical pathway. The first step is the binding of C3b to an activator. C3b is continuously generated in small quantities in the circulation, but in the free state, it is rapidly inactivated by factor H and I. Bound C3b, which is protected from such inactivation, interact with the factor B to form magnesium-dependent C3b. This complex is cleaved by factor D into two fragments BA and BB. The fragment BB remains bound to C3b and form C3bb complex, which is also called C3 convertase. C3 convertase is extremely labile and is stabilized by propelting. Stabilized C3 convertase cleaves more C3 and produce C3bb 3b complex, which is also called C5 convertase, which cleaves C5 into C5a, C5b. Now you know what happens next, because we have already studied in classical pathway the C5b initiate the formation of MAC, so-called membrane attack complex. So this is the initiation of alternative pathway by specific non-covalent binding of propelling to target membrane. And next is a lectin pathway. C4 activation can be achieved without antibody and C1 participation via the lectin pathway. Three proteins initiate this pathway named as MBL and MASP, MASP2, all present in the normal serum. We have introduced this MBL, MASP, MASP2 in the complement component materials, which is happened to say before. So MBL binds to the certain mannose residues on many bacteria and subsequently interact with MASP and MASP2. The MBL, MASP, MASP2 complex is similar to antibody C1 QRS complex, which is we have already said in classical pathway, and leads to the activation of C4, C2, C3. The rest follows as the classical pathway. So I have already said about this MPL, MASP, MASP2 when we have said about this complement components. MBL is, as we know, as you can see, MBL is an initiator complement component. Then MASP2 is enzymatic mediator. So by using all this, the last pathway, lectin pathway, will happen. So this is the end of the story. Thank you.